We have also done for the balance sheet of ATPs based on conditions. What conditions? Basing on that balance sheet is prepared, Nana. Number one, it starts always with glucose, isn't it?
Pose.
Kada?
Yes, sir, the only substrate.
Yes. Then no intermediate is withdrawn. Yes. And no others enter as intermediates also. Kada? ఎవరిది నాన్న ఇది అయ్యో ఇంట్లో వీడియో వచ్చేసి కెమెరా తిప్పుతుంటేనా సరే బట్ కదా
Yes, sir.
then also all these steps occur in that sequence. All 4 steps occur in that particular sequence. Then NADH of glycolysis enters mitochondria and takes part in ETS. Suppose if that doesn't enter, then 6 ATPs will be less, isn't it, Nana?
Yes.
Okay. So that part we have done yesterday. Then energetics done yesterday, right?
Yes.
Then, of course, we also have done the difference of oxidative phosphorylation and photophosphorylation. Yes? Yes or no?
Sir, we haven't like written about it. We just orally told.
Achha, okay. Better write, no. Aerobic, anaerobic, no need to write, but distinguishing oxidative and photophosphorylation better, Nana. So oxidative phosphorylation. Photophosphorylation. Okay. Of course, this is during respiration. This comparative analysis would have been better after completing photosynthesis, but still, with the idea you have about photosynthesis, there won't be any problem. So, seen during respiration. What about this, Nana?
Seen during photosynthesis.
Yes, easy. This will be in presence of oxygen. What about this, Nana? Photophosphorylation. In presence of what? Tell me.
Sunlight.
Sunlight. Then this occurs in mitochondria. What about this, Nana? Nana, chloroplast. Occurs in chloroplast. Occurs in mitochondria. Occurs in mitochondria of, you can say, all living eukaryotic cells. Mitochondria is the case with eukaryotic cells. What about prokaryotes? In the mesosomes of prokaryotic cells. Whereas this occurs in the chloroplast of, is it all living cells, Nana?
No, sir.
Green
Okay.
cells
Green.
of plants. And both are associated with ATP synthesis. So, oxidative in presence of oxygen, photo in presence of sunlight. This is during respiratory ETS, that is during photosynthetic ETS. So, during respiratory electron transport system, during photosynthetic electron transport system. But what is the similarity of both? That is important. In both the processes, ATPs are synthesized during translocation of protons across the membrane. Translocation of protons across the membrane. If it is respiratory, which membrane was it? In the mitochondria, inner membrane. What were the infoldings of inner membrane called, Nana? Crystic.
Crystal.
Yes. Why? Protons are channeled from intermembrane space to matrix side. We were getting ATP. So across the membrane, same thing in photosynthesis also, but that is across the membrane of thylakoid, chloroplast, granathylakoids. We know the structure of the chloroplast. So when here protons are channeled from lumen of thylakoids into stroma side, ATP are synthesized. Here ATP in the matrix, here ATP in the stroma. Protons reservoir here, lumen of thylakoids. Protons reservoir here, intermembrane space. What was that also called, Nana? Intermembrane space?
Lumen.
Perimitochondrial.
Yes. This is lumen of thylakoids. Mitochondria. See the similarity, Nana, in both while channeling of protons across the membrane. ATPs here are formed on matrix side. ATPs here are formed on stroma side. In the stroma, those ATPs will be used in dark phase for reducing carbon dioxide into carbohydrate. So in both of them, protons reservoir is there. In one it is intermembrane space, in another it is lumen of thylakoids. And ATP's reservoir in a mitochondrion is mitochondrial matrix. Next, let us see Antibolic Pathway. Okay to rub off up to this, Nana?
Yes, sir.
Okay. What is meant by amphi? Can you tell me?
Both.
Dual.
Two. Amphi, diplo, di. Meaning both. Amphistomatous. Meaning what, Nana?
Both.
Tomato on both
sides.
Stomata were equally on both sides. Amphitricus bacteria. What was, Nana? Bacteria with flagella at both ends. Amphitricus. Two tufts, one each at opposite poles. So amphi, diplo, di, meaning same. So here it means respiration is called amphibolic pathway because it involves one, breakdown process to release energy. Of course, this is main process. Also, synthetic process. Involving biomolecules formation from intermediates, various intermediates. It is significant not only in releasing energy, but also forming various kinds of molecules. Some of the intermediates of it, the carbon skeleton formed during this time is withdrawn, used for other biomolecules formation. Some examples we take. Done all of you this? Shall I rub off, Nana?
Yes.
Yes, sir.
Other than this, wherever phosphorylation, we were calling it a substrate level phosphorylation. Remember, Nana, why the substrate level phosphorylation I told you? Where energy released Energy and phosphates are from substrate itself. So their ATPs are not during translocation of protons. During translocation of protons is this. Okay, that's different. Now, coming to antibolic pathway, it is called so because it involves both breakdown process to release energy, that is mainly, and of course also synthetic process involving biomolecules formation from various intermediates. A few examples to cite. Example 1, alpha ketoglutarate acid. What is its importance, Nana?
Can be withdrawn to make glutamate and then protein.
Formed during TCA cycle. Of TCA cycle, alpha ketoglutarate acid of TCA cycle is withdrawn, can be withdrawn to form which amino acid, Nana?
Glutamate.
Glutamic acid for protein synthesis. Similarly, succinyl-CoA was intermediate of which step of respiration? Again of TCA cycle, formed during oxidation of alpha ketoglutarate acid into succinyl-CoA you got. So succinyl-CoA of TCA cycle can be withdrawn all rings of chlorophyll structure. Chlorophylls exhibit porphyrin structure with distinguishability into tetrapyral head and phytol tail. Isn't it, Nana? Who is this Bill? Governor Levy? 943. BILH. Who is that, beta?
Sir, it is Bilhan.
Bilhan. Okay. Okay, Nana. Charya. So, succinyl-CoA of TCA cycle can be withdrawn to form pyrrole rings of chlorophyll. A chlorophyll molecule structure, if you see, it is with tetrapyrrole head, Nana, with 4 pyrrole rings attached to single magnesium atom. While talking about magnesium, we were talking about in the mineral nutrition chapter. While talking about magnesium, we said associated with chlorophyll,
Ring structure of chlorophyll.
transfer, yes, processes. Then tail of carbons and hydrogens there. So this part is tetrapyrrole head. And this part is phytol tail. Together called porphyrin structure. This is chlorophyll exhibiting porphyrin structure. Porphyrin, pH1. Four pyrrole rings form the head part, then phytol tail, chain of carbons and hydrogens. So succinyl-CoA can be withdrawn. In which case, anabolism we call it. Similarly, acetyl-CoA can be withdrawn to form various biomolecules which are useful to the plants. Various biomolecules like ABA, which is a phytohormone, abscisic acid that causes closure of protects plants from water stress conditions by minimizing transpiration. Similarly, also used in gibberellins, which are also phytohormones. Similarly, cutin formation that forms cuticle. Okay. Similarly, phytol tail formation. Acetyl-CoA is used in so many ways. So all these constitute anabolic processes linked to catabolism. All these are anabolic processes linked to catabolism, respiration. Similarly, respiratory substrates, when you talk about respiratory substrates, can enter pathway as intermediates also. So catabolism involves respiratory substrates entering the pathway in different forms. Example one, for example, if you take lipids break into glycerol and fatty acid. Do you know fatty acids and glycerol together called what, Nana? Glyceride.
Glycerol.
With two fatty acids, diglycerides. With one fatty acid, monoglycerides. With three fatty acids, triglycerides. Then glycerol of these enters respiration as GAP, which is otherwise called P-gal. When fructose 1,6-bisphosphate undergone cleavage, you are getting a DAP and a GAP. GAP is otherwise P-gal, phosphoglyceraldehyde, nothing but glyceraldehyde phosphate, nothing but E3P, glyceraldehyde 3-phosphate. It will enter as this through DAP. dihydroxyacetone phosphate. Fatty acids enter as acetyl-CoA. Fatty acids convert into acetyl-CoA, then they will start with TCA cycle because they are forming acetyl-CoA that can be intermediate for TCA cycle. Lipids if it is. Proteins if it is how? Let us see.
So what is phytoalkane formation?
Tail part of the chlorophyll structure, Nana. So tail part formation time, acetyl-CoA is significant. Tail part is nothing but chain of carbons and hydrogens. Just remember better, we don't need that mechanism right now. Second, if it is proteins, break into amino acids and take part in respiration. As pyruvic acid, acetyl-CoA. Acetyl-CoA is common for all kinds of substrates you are noticing. For lipids, it was through acetyl-CoA there, proteins, acetyl-CoA there, carbohydrates, anyhow we know through acetyl-CoA. Or even as alpha ketoglutarate acid. How it is, we have seen under nitrogen metabolism chapter also. Glutamate losing amino group to form other amino acids will become alpha ketoglutarate that can again enter respiratory pathway. So now flow chart form, go to fresh page now. Same way, pyruvic acid can be withdrawn to form proteins. Acetyl-CoA can be withdrawn to form proteins. Ketoglutarate acid can be withdrawn to form proteins. So both ways it is catabolism, anabolism. You are going to see. Done, Nana?
Sir, 1 minute, sir.
No problem. Take no problem.
Yes, sir, done, sir.
Okay, very good. If it is carbohydrates, we started. Let us see. So go to fresh page, write the flowchart form, Nana. Can be the substrate, we know it. Lipids can be the substrates, we know it. Proteins can be the substrates. Even organic acids also can be the substrates. If it is carbohydrates, like starch, hydrolyzed into what, Nana? Glucose and fructose. By what enzyme was it? Remember? Starch hydrolyzed into glucose and fructose.
Invertase.
Invertase, very good. So glucose was undergoing glycolysis part, giving rise to fructose 1,6-phosphate, and that was split into a dap and a gap, which is also called PGAL, continuing with remaining reactions, giving what? Pyruvic acid, which then was giving what? Acetyl-CoA, and this was involved in condensation with oxaloacetic acid to form what, Nana? Citric acid, first stable compound, undergoing a series of reactions forming alpha keto glutaric acid, succinyl-CoA, succinic acid, fumaric acid, malic acid, regenerating way. If it is proteins, they break down into amino acids, okay, no, amino acids can convert into pyruvic acid to take part here, can convert into acetyl-CoA to take part here, can convert into alpha ketoglutaric acid to take part there. Proteins can enter as pyruvic acid, as acetyl-CoA, as alpha ketoglutaric acid. Lipids, if it is, they are hydrolyzed into fatty acids, and glycerol. What was glycerol? Remember, Nana? Trihydroxypropane, isn't it? Glycerol will enter as gap through DAP. Fatty acids will enter as acetyl-CoA. See? They are entering somewhere in the middle. If it is organic acids, of various types can enter as malic acid, succinic acid, fumaric acid, etc. So these indicate catabolism. Breakdown process. If reverse, it is alpha ketoglutarate acid withdrawn to form amino acid, then proteins. Same is the case with these also. Same is the case. Then what do you call the reverse, Nana? Anabolism. Forward, catabolism. Upward, anabolism. So, because of its significance in both catabolism and anabolism, it is called amphibolic pathway. Done, Nana? Even these can be withdrawn to form various organic acids. Same is the case with this. Same is the case with this. Reverse anabolism. Forward catabolism. Done?
Done, sir.
Okay. Next is the last aspect under respiration. RQ value, respiratory quotient. RQ, as well called respiratory ratios. Is nothing but the ratio of volume of CO2 released during respiration. CO2 evolved or released to the volume of O2 utilized. Is called RQ value. RQ value, it is measured by respirometer, which is used even for measuring respiration rate also. RQ value helps in finding out 1. Type of respiration. 2. Nature of substrate taken part in respiration. Based on RQ values, you can find out what type of respiration occurred and which type of substrate involved. So RQ value literally varies depending on what, Nana? Nature of substrate and type of respiration. So number 1, RQ value for carbohydrates. First, let us see. In aerobic respiration, if it is carbohydrates, no, it starts with glucose. If it is so, equation goes this way: C6H12O6 required to be supplied with 6 molecules of O2, giving rise to making out the product easy. And once you know the substrate molecules number, carbon dioxide equal to the number of carbons here, so it should be 6 molecules of CO2. Then of course 6H2O. Number of hydrogens divided by 2 will give you number of water molecules you are going to get. Easy to make out. Then of course, energy. So RQ value to be calculated is volume of CO2 released to volume of oxygen used. It is nothing by 6 by 6 is equal to 1.
1.
So here RQ value is a unity. Therefore, RQ value for carbohydrates in aerobic respiration is a unity in terms of mathematics, we say. What can be the example here? Germinating cereal grain seeds. Germinating seeds of cereals which are rich in carbohydrates, like rice, it may be, maize, it may be, wheat, it may be, which are generally said to be rich in carbohydrates. If it is so, same carbohydrates under anaerobic conditions. Next heading. Do you see any oxygen used in anaerobic respiration, Anna? No oxygen utilized. So RQ value,
Infinity.
therefore, is infinity. Anything divided by 0 is infinity. What is the symbol for this, Nana? So C6H12O6 giving rise to, let us take it as 2 molecules of C2H5OH plus 2 molecules of CO2. Two by 0, infinity. RQ value for same carbohydrates if it is succulent xerophytes. In case of succulent xerophytes, no CO2 evolved, especially during daytime because of the stomata closure. Then what do you take, Nana? 0. 0 divided by anything should be 0 only. For carbohydrates, in succulent xerophytes, it is 0 because no CO2 evolved. So 0 divided by anything will be 0 only. Then important is this again. RQ value for lipids. Generally, it is less than 1 because lipids have less O2 and need naturally more O2. When they are least or poor in oxygen, so they require to be supplied with more oxygen. Example, tripalmitin. Don't compare or confuse with palmitic acid. Remember, Nana, palmitic acid was a fatty acid with how many carbons? 16 carbon fatty acid.
Including the carboxyl.
Including that of carboxyl. Very good. What about arachidonic acid?
20
20 carbon, including
carboxyl.
Of carboxyl. This is tripalmitin.
48 carbons.
One molecule, suppose if you take 51 carbons, better it is. 2 molecules of tripalmitin at a time are involved actually. So 2 molecules of C51 H98O6 requires to be supplied with 145 O2s. Once you know this and this, easy to make out the product, Nana. Equal to the number of carbon atoms is carbon dioxide. So 102 molecules of carbon dioxide. Then half to the number of hydrogens is water. 98 into 2.
49.
No, 98 into 2. Divided by again 2. So stay
98.
water molecules. Finish. Automatically equation is balanced. So RQ value is equal to 102 divided by 145 comes to 0.7 nano, which is less than 1. For proteins also, no need to go with equation there, just to remember. Yeah, we're finishing. RQ value. Here example can be germinating castor seed, rich in fats. RQ value for proteins is also less than 1, around 0.8, because poor in oxygen to carbon ratios. Example, germinating bean seed. You can take it as.
Sir, is it 0.9?
0.8, Nana. 0.8 to 0.9 actually. For proteins is concerned. Or better you write the range otherwise, no problem. Okay, Nana. So the chapter respiration is done. Tomorrow we go with growth and development. Only few aspects are there in that. Up to conditions of growth, which need one class, hopefully tomorrow. I think we stop here. Time also.
Thank
Yeah.
you, sir.
11:15. Yes, Nana. So bye. See you tomorrow. Those who have done can leave better.
Good morning, sir.
Good
Thank you,
morning,
sir.
juniors.
Sir,
Bye.
you
Sir,
will
should we
again
join
start the
again?
meeting.
Good morning.
Wait, juniors. I'll close it and come back better.
Okay, sir.
Thank you, sir.
Bye, Nana.